Hello students, welcome to readmedprep Academy channel. Today in evolution part 2 we are going to discuss about biological evolution and evidences for biological evolution. The formation of protobionts. Abiotically, molecules can spontaneously self-assemble into droplets that enclose a watery solution and maintain a chemical environment different from their surroundings. Scientists call these spheres as protobionts. How the protobionts are formed? The macromolecules get accumulated and a membrane component is formed around it. These protobionts slowly acquire the food and they undergo metabolism and self-maintenance. And these protobionts can further undergo self-replication and reproduction. Here you can see a protobiont which has macromolecules inside with surrounded semi-permeable membrane made up of polypeptide. Sometimes they acquire the nucleic acids like the RNA and the DNA. They undergo primitive metabolism in a protected environment. Protobionts are predecessors of prokaryotic cells. They are aggregates of abiotically produced molecules. They can form by self-assembly. They cannot reproduce. They can maintain an internal chemical environment. They acquire food substances like the glucose phosphate, which undergoes metabolism inside the protobiont. And these chemicals undergo simple metabolism and they help in the release of maltose and phosphates. What are liposomes? Liposomes are lipids in a solution that can self-assemble into a lipid bilayer. Some of the proteins inside the liposomes acquire the properties of enzymes, resulting in fast multiplication of molecules. Liposomes, the evolution of the science and technology, has been used in the development of drug carrier concept as a promising delivery system. The liposome was adopted as a promising delivery system because its organized structure, which could hold drugs depending on their solubility characteristics in both the aqueous and lipid phases. In the image you can see the micelle and a molecule of liposome which is made up of a bilayer sheet. What are coacervates? Coacervates with nucleoprotein and nutrients had a limiting surface membrane that had the characters of a virus or free living genes. Subsequently, number of genes united to form protoviruses, somewhat similar to the present day viruses. Here in this image you can see that the ocean is considered as a primordial soup in the prebiotic earth, and the primordial soup contained lot of inorganic substances. The coacervate droplets were formed by the assimilation of nucleic acid and amino acid as well as with fatty acids, and inorganic and organic chemicals got accumulated inside a cell membrane. The interacting coacervates occurred which causes interaction with the surrounding environment. They exchange substances from within to outside and from outside to within. What are the differences between coacervates and probionts? Definition: coacervates are droplets formed from charged polymers. Protobionts are spherical microspheres of diverse organic and inorganic compounds enclosed by the lipid membranes. Coacervates were considered as a kind of protocells, whereas protobionts were the evolutionary precursors of prokaryotic cells. Coacervates, the membranes are single, and in protobionts they have a double membrane. Cell types. There are two major cell types that appeared during this time were significant: Monera and Protista. One form of the earliest cell contained clumps of nucleoproteins embedded in the cell substance. Such cells were similar to the Monera. They are considered as the ancestor to the modern bacteria and blue-green algae. You can see in the image the assimilation of the nuclear material in the center surrounded by the cell wall and the cell membrane. Protista, which is another cell type, the other form of earliest cells that contain nucleoprotein clumps that condensed into a central mass surrounded by a thin membrane. This membrane separated nucleoproteins from the cell substances. Such cells were referred to as Protista. Now, evolution of Protista. A prokaryote ingested some aerobic bacteria. These aerobes were protected by the prokaryote and produced energy for it. B, you can see over a long period of time, these aerobes became the mitochondria and they couldn't live on their own anymore. So the cell containing the mitochondria developed into the animal cell, which has to depend on others for food. In C, you can see some of these primitive prokaryotes also ingested cyanobacteria. Cyanobacteria contain photosynthetic pigments. In D, you can see over a long period of time, the cyanobacteria in these prokaryotes became the chloroplasts and couldn't live on their own anymore. And this led to the development of the plant cell, which contained the mitochondria for respiration and the chloroplast for photosynthesis. This is how the Protista evolution occurred. Food procurement. When the natural sources of food in the ocean declined in the course of time, the ancestors of Monera and Protista had to evolve different methods for food procurement. The types of food procurement: parasitism, where one organism had to depend completely on other organism for food. Saprophytism, where the organisms depend on the dead and decaying organic matter for food. Predator or animalism, where one animal killed and ate the other animal for food. Chemosynthesis of photosynthesis, where chemical substances or the source of sunlight was used for the synthesis of starch and other food materials. Photosynthesis and oxygen. When the number of photosynthetic organisms increased, there was an increase in the free oxygen in the sea and atmosphere. The atmospheric oxygen combined with methane and ammonia to form carbon dioxide and free nitrogen. You can see in this equation CH4, methane combined with 2 molecules of oxygen to give carbon dioxide and 2 molecules of water. The second equation, you can see 4 molecules of ammonia combined with 3 molecules of oxygen result in the formation of release of nitrogen 2 molecules and 6 molecules of water. The presence of free oxygen brought about the evolution of aerobic respiration, which could yield large amounts of energy by oxidation of foodstuffs. Thus, prokaryotes and eukaryotes slowly evolved. In this image, you can see that the plants absorb water through the roots, and these water and the nutrients reach the leaves, and the leaves, by utilizing the sunlight and from the absorption of carbon dioxide, released oxygen and water vapor into the atmosphere, and this helped in the synthesis of food called photosynthesis. And these plants, when they release oxygen, this oxygen was consumed by animals. These animals, by respiration, release a lot of carbon dioxide into the atmosphere, which is consumed by the plants, and the cycle goes on. Experimental approach to the origin of life. Uray and Milner in 1953 paved way for understanding the possible synthesis of organic compounds that led to the appearance of living organisms. Uray and Milner experiment. In their experiment, a mixture of gases was allowed to circulate over electric discharge from a tungsten electrode. A small flask was kept boiling, and the steam emanating from it was made to mix with the mixture of gases like the ammonia, methane and hydrogen in a large chamber that was connected to the boiling water. The steam condensed to form water, which ran down through the U-tube. An experiment was conducted continuously for a week, and liquid was analyzed. This liquid contained glycine, alanine, beta-alanine and aspartic acid. Thus, Milner's experiments had an insight as to the possibility of abiogenetic synthesis of large amount of variety of organic compounds in nature from a mixture of simple gases, in which the only source of carbon was methane. Later, in similar experiments, formation of all types of amino acids and nitrogen bases were noticed. Here in this image, you can see the diagrammatic representation of Uray-Milner's experiment, where a flask with water is taken and continuously boiled. Steam that emanates passes through a large bulb which has a tungsten electrode which shows spark discharge, and the flask also contains gaseous mixture containing the methane, ammonia, hydrogen and water vapor, and this condensation results in formation of aqueous medium containing organic compounds, and excess of heat is condensed by a condenser. and the accumulated aqueous medium is collected in a flask and was analyzed to found organic compounds. Now let us move on to the evidences for biological evolution. The evidences are paleontological evidences, evidences from comparative anatomy, embryological evidences, and molecular evidences. Let us discuss one by one. Paleontological evidences. Paleontology is the study of prehistoric life through fossils. Fossils are described as the true witnesses of evolution or documents of various geological strata of the evolution. What is fossilization? It is the process by which plant and animal remains are preserved in sedimentary rocks. They fall under three main categories: actual remains, petrifaction, natural molds and casts. Actual remains. The original hard parts such as the bones, teeth or shells are preserved as such in the Earth's atmosphere. This is the most common method of fossilization. When marine animals die, their hard parts such as bones, shells, etc. are covered with sediments and are protected from further deterioration. They get preserved as such as they are preserved in vast ocean. The salinity in them prevents decay. The sediments become hardened to form definite layers or strata. In the image you can see the fossil of starfish. For example, woolly mammoth that lived 22,000 years ago were preserved in the frozen coast of Siberia as such. Several human beings and animals living in the ancient city of Pompeii were preserved intact by volcanic ash which gushed out from the Mount Vesuvius. What is petrifaction? When animals die, the original portion of their body may be replaced molecule by molecule by minerals and the original substance being lost through disintegration. This method of fossilization is called petrifaction. The principal minerals involved in this type of fossilization are iron pyrites, silica, calcium carbonate and bicarbonates of calcium and magnesium. In the image you can see the calcified deposits in the snail fossil. What are natural molds and casts? Natural molds and casts, even after disintegration, the body of an animal might leave indelible impression on the soft mud, which later becomes hardened into stones. Such impressions are called molds. You can see in the image on the right side the external mold and an unaltered shell as a fossil. The cavities of the molds may get filled up by hard minerals, get fossilized, which are called casts. The hardened fecal matter, termed as coprolites, occur as tiny pellets. Analysis of the coprolites enables us to understand the nature of the diet the prehistoric animals thrived on. Now, what are the evidences from the comparative anatomy? Similarities in structure between groups of organisms are accepted as indicators of relationship. For example, a comparative study of the forelimbs of different vertebrates exhibits a fundamental plan of similarity in structure. These relationships can be studied under homologous organs, analogous organs, vestigial organs, connecting links, and atomistic organs. Homologous structures. In vertebrates, comparative anatomical studies reveal a basic plan in various structures, such as the forelimbs and the hind limbs. Forelimbs of vertebrates exhibit anatomical similarity with each other and is made of similar bones such as humerus, radius, ulna, carpals, metacarpals, and phalanges. Here in this image, you can see the bones of the wing of bird, bat, whale, cat, horse, and humans. All these mammals share common bones like the humerus, the ulna, the radius, the carpals, and the phalanges. Structures which are similar in origin but perform different functions are called homologous structures that bring about divergent evolution. Similarly, the thorn of bougainvillea and the tendrils of cucurbita and Pisum sativum represent homology. The thorn in former is used as a defense mechanism from grazing animals, and the tendrils of the latter, like the cucurbita and the Pisum, is used as a support for climbing. Here you can see the comparison of the thorn of bougainvillea and the tendril of cucurbita. Both are homologous structures. Now, analogous structures. Organisms having different structural patterns. but similar function are termed as analogous structures. For example, the wings of birds and insects are different structurally but perform the same function of flight and brings about convergent evolution. Here you can see the wing of a bird with the bone humerus, the radius and ulna, the carpals and the phalanges. Here you can see wings of the insect, the hind wing and the forewing, and the wings and the feathers perform the same function of flight. Other examples of analogous organs include the eyes of octopus and of the mammals and the flippers of penguins and dolphins. On the left side you can see the eye of the octopus. On the right side you can see the eye of the humans. The eye of the octopus shows the optic nerve, optic ganglion, the photoreceptor cells, the ciliary muscle, the lens, the iris and the nerve fibers. In the human, in addition, you can see bipolar cells and the blind spot. Analogous structures in dolphin, which is a mammal, you can see the flipper. The penguin, which is a bird, you can see the wing. In the shark, you can see the fin. The flipper, the wing and the fin in dolphin, penguin and shark respectively are analogous structures. Root modification in sweet potato and stem modification in potato are considered as analogous organs. Both of these plants have a common function of storage of food. Here you can see the analogous similarity between potato and sweet potato. The potato is an underground stem, whereas sweet potato is an underground root. Both are starchy food items. Now let us move on to the vestigial organs. Structures that are of no use to the possessor and are not necessary for their existence are called vestigial organs. Vestigial organs may be considered as remnants of structures which were well developed and functional in the ancestors, but disappeared in the course of evolution due to their non-utilization. Human appendix is the remnant of cecum, which is functional in the digestive tract of herbivorous animals like rabbit. Cellulose digestion takes place in the cecum of these animals. Due to change in the diet containing less cellulose, cecum in human became functionless and is reduced to a vermiform appendix which is vestigial. Here in this image you can see the human digestive system on the left side and the rabbit digestive system on the right side. In the human digestive system you can see the cecum is reduced and the vermiform appendix is vestigial, whereas in the rabbit digestive system you can see the cecum is well developed where the cellulose digestion occurs. Other examples of vestigial organs in human beings include coccyx, wisdom teeth, ear muscles, body hair, mammae in male, nictitating membrane of the eye, etc. In this image you can see the vestigial organs in the human beings. On the left side the nictitating membrane, the pointed canine, the third molar, the vermiform appendix and pyramidalis muscle. On the right side you can see the muscles of the ears to move them, hair on the body, mammae on the male, segmental muscles on the abdomen and caudal vertebrae. Organisms which possess the characters of two different groups, which is called the transitional stage, are called the connecting links. Example, peripatus, which is a connecting link between Annelida and Arthropoda. You can see the peripatus in the image where it has a antenna, eyes, oral papillae, brain, gut, skin, leg muscles, slime glands and tubular heart. It is a connecting link between Annelida and Arthropoda. Here you can see Archaeopteryx, which is a connecting link between the reptiles and the apes. When you examine the bird, you can see reptilian character. Tooth beak, which is a reptilian character. Wing claw, reptilian character. Airfoil wing with contour feathers, which is an avian character. Long tail with many vertebrae, which is a reptilian character. Atomistic organs. Sudden appearance of vestigial organs in highly evolved organisms is called atavistic organs. Example, presence of tail in the human baby is an atavistic organ. In the image you can see some atavistic organs like the dense hairs, the tail in humans and the large canines. What are the embryological evidences? Embryology deals with the study of the development of individual from the egg to the adult stage. A detailed study of the embryonic development of different forms makes us to think that there is a close resemblance during development. The development of heart in all vertebrates follows the same pattern of development as a pair of tubular structures that later develop into two-chambered heart in fishes, three-chambered in amphibians, and in most reptiles, and four-chambered in crocodiles, birds, and mammals, indicating a common ancestry for all vertebrates. Here in this image you can see the heart's anatomy, where two-chambered heart in fish. In amphibians it's a three-chambered heart. In reptile, birds and mammals it is a four-chambered heart. Hence scientists in the 19th century concluded that higher animals during their embryonic development pass through stages of lower animals or ancestors. Ernst von Haeckel propounded the biogenetic law or theory of recapitulation. It states that the life history of an individual, ontogeny, briefly repeats or recapitulates the evolutionary history of the race, phylogeny. In other words, ontogeny recapitulates phylogeny. The embryonic stages of a higher animal resembling the adult stage of its ancestors. Appearance of pharyngeal gill slits, yolk sac, and appearance of tail in the human embryos are some of the examples. The biogenetic law is not universal, and it is now thought that animals do not recapitulate the adult stage of any ancestors. In this image you can see the embryonic development of vertebrates, where the gill slits become the pharyngeal pouches and later the lungs in the humans, and the gill slowly disappears, and there is development of forelimb buds and the hindlimb buds in the humans, and the tail that was present in the fishes slowly disappears and is absent and becomes vestigial in man. The human embryo recapitulates the embryonic history and not the adult history of the organisms. The comparative study of the embryo of different animals shows structural similarities among themselves. The embryos of fish, salamander, tortoise, chick and human start life as a single cell, the zygote, and undergo cleavage to produce blastula, change to gastrula, and a triploblastic mass of cells. This indicates that all the above said animals have evolved from a common ancestor. Here in this you can see the embryological evidences in fish, salamander, tortoise, chick and human, where the gills slowly become the pharyngeal pouches and the lungs in humans, and the post-anal tail, which was present in the lower animals, slowly disappears and becomes vestigial in humans. What are the molecular evidences? Molecular evolution is the process of change in the sequence composition of molecules such as DNA, RNA and proteins across generations. It uses principles of evolutionary biology and population genetics to explain patterns in the changes of molecules. One of the most useful advancements in the development of molecular biology is proteins and other molecules that control the life processes are conserved among the species. A slight change that occurs over time in these conserved molecules, DNA, RNA and protein, are often called molecular clocks. A molecular clock, when you take, the species that diverged recently showed only few nucleotide differences, whereas the species that diverged a long time ago have many nucleotide differences. You can see in the image where the common ancestor having a nucleotide sequence and the change is very single, whereas as evolution evolves about 50 million years ago, the changes become more and the nucleotide sequences change in a more numbers. Molecules that have been used to study evolution are cytochrome C, respiratory pathway, and ribosomal RNA in protein synthesis. The genes of many organisms show important similarities at the molecular level. Similarities in DNA can be used to help determine classification and evolutionary relationships. The more similar the DNA sequence of two species, the more recently they shared a common ancestor, and the more closely related they are in evolutionary terms. In the right set table you can see the cytochrome C evolution, the organism and the number of amino acid differences from humans. Chimpanzee 0, rhesus monkey 1, rabbit 9, cow 10, pigeon 12, bullfrog 20, fruit fly 24, wheat germ 37, yeast 42. So this is how the evolution of cytochrome C occurred. So today in evolution part 2, we discussed about the biological evolution and evidences for biological evolution. So thank you. Kindly subscribe, like, share and comment our channel Read MedPrep Academy. Register for UG and PG NEET-like MCQs in our website www.readmedprepacademy.com. Our Facebook ID is readmedprep academy. Our email is readmedprepacademy@gmail.com. Our Instagram is readmedprep academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Join Read MedPrep Academy for preparing for NEET exams. Kindly WhatsApp the number given below. Thank you very much.